So this is a small talk that is titled Structural Dynamics of Neural Clusters. So we're going to go through a review of neural architecture, the way it's been thought of for about 100 years. And then we're going to look at neural cluster formation, also classical concepts. Basically, like Ramji said, from the neuroscientists and the neuroanatomists, because that's where this idea of a cluster began from looking at the microscope. Then we're going to take a very fast tour through new data. So we have mirror neurons, power loss, subcortical processing, protein folding, engrams, and DNA encoding. And we have to see whether we think that this data is real and whether it should be incorporated into any new understanding of the term neural cluster. So that's what section 4 will be, neural cluster formation and dynamics, that sort of incorporates other data that have come in. And 5, I'm going to take a stab at this new term called a neural syntax, which is the underlying algorithm that the brain applies to understand and interact with all information, whether it's incoming or returning up from memory stores. So we have a classical anatomical picture here on the left side of a neuron. So we have the soma, the cell body with the nucleus and the mitochondria, the endoplasmic reticulum, and others that help the soma live. And then we have the dendrites, which are these feelers that the that the soma sends out to neighboring cells that collect information. So those are the inputs to the cell. And then there's the output, which is the axon, which goes out and connects either to other cells or other muscle cells, so other neurons or other muscle cells, to things called synaptic boutons. So that was the traditional view for a very long time, but it turns out that the axon is not the only way for a neuron to send an output. It also sends non-synaptic outputs through gap junctions. So non-synaptic communication is a thing. But we still don't know the full impact of that or all of the full details of that, except that the consensus is that non-synaptic communication is orders of magnitude slower than synaptic communication. But it does travel longer. So it's slower, but longer distance. So on the right side, we have a section of a neuron. And you can see that the neuron isn't just surrounded by other neurons or living in splendid isolation. It is surrounded by a huge amount of supporting cells, and those are oligodendroglia and other glial cells, microglia, astroglia, satellite cells, and so on. And they are engaged in a 24/7 communication with the neuron. So the support is continuous throughout life. And they nourish the neuron and they remove the neuron's wastes. And so as I was telling Ramji, one of the cells that I've been looking at for some years is the cell called a pericyte, which splays itself across capillaries. And this pericyte also is in direct communication with neurons. It's not in this cartoon. And that also helps remove wastes from neurons, especially a protein called the alpha-synuclein protein, which is kind of important. So we have mitochondria inside all of these cells that provide energy for the neurons to work and to generate its action potentials. So what's an action potential? Well, we have a synapse. So that's the bluish green area on the upper side of this picture. And then we have neurotransmitters that are released from vesicles inside these synapses. And they travel across a gap junction into either a dendrite of a neighboring neuron or a muscle cell, and they generate a calcium spike. And that triggers an electrical signal, that a potential that flows down the receiving cells. And what the neuroscientists have discovered, over many, many years is something called long-term potentiation. So what happens if you keep stimulating a neuron, over time, it starts generating more and more neurotransmitters and stores them inside the vesicles and the terminal boutons. And then each spike that you generate in that neuron releases more neurotransmitters into the synaptic cleft than before. And the term for that is potentiation. So you have potentiated that nerve into sending more neurotransmitters and causing bigger spikes and more spikes. And so the neuroscientists for many years thought that this was the basis of memory storage, that the potentiated neuron stored memories. And then they discovered other types of cells that they called engram cells. And they postulated that this mechanism of long-term potentiation caused the storage of memories. So you have more dendrites connecting to cells, and the more dendrites you have connecting to cells meant that it was a cell that stored memory. So more dendritic connections meant memory. And that's what this picture shows. So they took a rat and conditioned it with fear, and they looked at the number of dendrites. The number of dendrites mushroomed, and they said, look, it's memory being formed before our eyes. Around the same time, Donald Hebb said, well, one nerve, one neuron is all very interesting, but what's happening, outside of each cell? And he postulated that a nerve assembly exists, which is a group of cells that generate a spike at the same time in response to a stimulus. So when a stimulus comes in, it's not just one neuron that fires, it's a bunch of neurons that fire. But his, real good interpretation was that these neurons aren't just in one particular location. They're not in geographic proximity with each other. They can be distributed all over the brain. And that's what this picture shows. So you have the green assembly, and those cells are all around. And then you have the red assembly, and you have the blue assembly, and the purple assembly, and so on. And so they fire in response to different sort of stimulations. And that was the other aha moment. So we do have cells that fire in response to the color yellow. We have cells that respond to certain shapes, certain orientations. So there are cells that respond selectively to slanted lines as opposed to straight lines. They do exist. So this is not a bad idea. We do have collections of cells across the brain that respond to selective input. So that was a significant advance. And so, he came up with this thing called plasticity, cells that fire together, wire together. And that's been sort of a dogma ever since, that this is one of the mechanisms of learning. Then if you look at the spikes of an individual neuron, you find out that it's not just an individual neuron, it's also regional potential called a local field potential. And then occasionally you find that it actually becomes a whole brain potential. So this is something that is found in all mammalian brains, regardless of size. And it's very interesting. The increase in size that you see on this lower picture here on the right side, it doesn't matter. The size actually doesn't matter at all to the dynamics of the nerves firing together. So you have the same wave patterns in the smallest mammalian brain as you have in the biggest, which is the human brain. And they have the same pattern on EEGs. So you have delta waves, which are the slowest waves, 0.1 wave per second to 3 waves per second. And then you have theta, which is somewhat faster, alpha, which is what we call the normal waking alert rhythm. And then you have beta waves, which are faster waves, usually seen in people who take benzodiazepines. And then you have gamma waves, which are 30 to 100 hertz, really fast waves. And gamma waves, you would think maybe are seen only in cases of pathology, but no, they actually exist in daily life as well. And like I said, they're evolutionarily conserved, which means they're important. So we do have to take their existence into account when we think about how the brain functions. Which brings us to the second part, which is the neural cluster. So when people looked at dissections of the brain under the microscope, they discovered 100 years ago, through the work of a neurologist called Ramon E. Cajal, and they developed staining techniques to stain the individual nerves and the axons and the dendrites and so on. And they found something interesting. They found this columnar pattern. So the picture on the right side is just the top 2 millimeters of the cortex of a brain. This is actually a rat cortex. But if you see, the columns are discrete. They are not entirely connected to each other, though there are cross-columnar connections. But the fact that they are columnar is very clear. You cannot deny it. And they also have layers within the columns. And so we were able to delineate 6 cortical layers in the neocortex. And we found 3 layers in the archicortex, or the amygdala, or the hippocampus. And so the architecture of this was thought to be important in the way the brain functions. The original idea had been that all the information that comes into the brain shoots up like a water fountain to the topmost layer, layer 1, and then it percolates down the column to the lower layers. So it's one column, information comes at the top, and then it percolates down, meaning it's processed as it percolates down. And then there's an output that's generated in layer 6, which enters the myelinated neurons, the so-called white matter of the brain. And for many decades, that's how it stood, that this was how the brain processed information. So it also had clinical implications. So for example, we see here on the lower left side, a transverse picture on the left, which shows the columns quite clearly. You can see these deep blue lines with the cell bodies in red in between. But on the right side, you have a top view. It's sort of like looking at the forest from above, from a helicopter or drone, and you see that you have these columns that you can see on end. And so neurosurgeons thought some decades ago, 30, 40 years ago, when we have people with intractable seizures, intractable epilepsy, what happens if we make these 2 millimeter cuts around each column and isolate each column so that the bad column, the rotten apple in the bushel, can spike all it wants, but it won't spread and become a generalized seizure. And so that was a standard technique for many, many decades. And you can see on the right side a classic EEG pattern from a patient with petit mal or absence seizures. So it's a 3 Hz spike in wave. So each of the vertical lines represents 1 second of time. And you can clearly see that the brain wave, which was normal on the left side, the left half, each area of the brain, each region of the brain has its own thing. But suddenly the seizure begins. The electrical activity, spiking activity spreads across the entire brain, and the entire brain starts spiking at 3 Hz. And generally people have what's called an absence or a pause. They just basically freeze. Nothing comes in, nothing goes out. And so they become absent, which is what the original term meant. So the 2 millimeter cut, surgeries were actually effective in some people. Then the neuroscientists and the EEGers also studied the larger waves, the whole brain waves. And so we found that there's these interactions between waves called cross-frequency coupling. And some people thought that that was the holy grail, the way the brain puts together words to make sentences and then create speech and language. So they came up with this thing called a theta gamma neural code. So if you have a high amplitude gamma wave within the time cycle, between the 2 peaks of a theta wave, then they call that a letter. They said this is, I think, the neuroscientific basis of letter formation. This is, we identify this as the brain forming a letter. And then on the right side, you have low theta gamma coupling. And there was some basis for it. So when you had regions with high theta gamma coupling, they also correlated with regions of larger long-term potentiation within the neurons in that region. So they coupled them together and said you see more long-term potentiation in areas with high theta gamma coupling. So it must mean something, and it probably means that either a letter is being formed or a memory is being stored. So here we have a newer way of looking at the same 6 cortical layers. On the leftmost side, you have the histological view, which is what you do with staining and looking at it under the microscope. All of this is within 2 millimeters from top to bottom. So white matter means bottom. That's where the axons leave. But then when you do very fine analysis using newer visualization techniques, with newer modeling techniques, like this picture from the Max Planck Institute, you find that the columns actually are rather more discrete than originally envisaged. So, you can see these clear layers on the stain, but when you look at it with other ways, it's not quite so clear. You have nerves, neurons, cells distributed throughout the 6 layers, and they're all connecting to each other, and they're all connecting to the rest of the brain. And so this sort of changed the idea that it's like a water fountain where the water shoots up to the top and then percolates in a linear unidirectional fashion downwards. And then 10 years ago, Randy Bruno and his team, proved that deep cortical layers are activated directly by the thalamus. So what's the thalamus? The thalamus are a bunch of nuclei that are deep in the middle of the brain, deep in the center of the brain. They're extremely important cells. They've variously been described as the way station for information coming up from the spinal cord, to a distributor cap that distributes signals to different areas of the brain, some to the cortex, some to the cerebellum, some to the amygdala, and so on. But what Bruno and his folks discovered is that a column, which had been called a neural cluster, isn't a unitary column. So it turns out that the top layers of this anatomical column, which had previously been called a neural cluster, are innervated separately from the lower portions of the column, and the lower portions get separate direct innervation from the thalamus, and the cortex gets separate direct innervation from the thalamus. And that was a thunderclap finding, because it got rid of this unitary water fountain theory, but it also raised the possibility that you finally understood why the cortex and the deeper areas could act independently of each other. And you see that clinically. We've seen this clinically for decades. So we have patients with what's called blind sight. Blind sight happens in people whose cortex, so the top 2 layers, has been damaged either by a stroke or trauma, and they cannot see. And you ask them, what do you see? And they go, it's black. I don't see anything. They really can't see. But what is interesting is that their brain can still see. And so Bruno's discovery explained why this is. It's because the thalamus sends information to the lower areas of each column independently of the upper areas. So even when the upper areas stop working for some reason, the lower areas are still working. They're still receiving information. They're still processing it. And so you have this phenomenon called blind sight, where the brain sees, but the person does not. And they were able to chase that down into the thalamus, the lateral geniculate nucleus of the thalamus. So that's for someone who cannot see with both hemispheres. So what happens in people with unilateral neglect or hemi-neglect? Hemi-neglect is seen in people with large middle cerebral artery strokes that affect the parietal lobe. It's as if one side of the earth, one side of the space just doesn't exist anymore. So they neglect it completely. You ask them to draw a clock, and they'll, you put a circle, but they will put in all the numbers, all 12 numbers, only on one half of the circle. And sometimes it's permanent. Sometimes you can rehabilitate it quite a bit. But how do you explain hemi-neglect in people? They just don't have any awareness of one side of the space. So if their stroke is on the left side, on the right side just doesn't exist. They will only comb the left side of their head. They will only button the left side of their shirts and so on. The right side just does not exist at all anymore. But what is interesting is, according to their brains, the right side still exists. And so you have visual processing without awareness. So even in people with heavy neglect, their brain does pay attention to what's going on in the right side. If you throw a ball at their face from the right side, their hand will go up to block the ball. And you ask them, why did you do that? They'd be like, what did I do? Because it's on the right side. It doesn't exist. So again, this goes in with Randy Bruno's discovery that the lower portions of the columns are sort of independent of the upper portions of the columns. So this is something that we have to take into account when we come up with a newer understanding of the term neural cluster. So then there's something called face trait. So it turns out our brains are very good at analyzing faces, and they do it within a few milliseconds, and they decide whether or not this person appears trustworthy or untrustworthy. The brain decides whether or not that person looks happy or sad or afraid. This study here showed that the thalamus and the amygdala support evaluation of face trait in blind sight. So the people aren't seeing who is standing in front of them, but their brain can see that that person exists and that person looks scared. And so this has now been shown for some years in different teams. So that's another thing that we have to take into account. And then here's a very interesting and recent finding by Dr. Mariusz Pachitariu, who was kind enough to let me show you this video. So what we have here, what Mariusz has done is he has made a 10,000 cell array. So an array is a sensor. So he's built a sensor that he can put on the surface of a brain, in this case, a mouse, and it records the spiking of 10,000 nerves at the same time. And so what we are seeing through his array is what the mouse's cortex is seeing in real time. And what the mouse's cortex is seeing in real time isn't just information coming up from the mouse's eyes. And that was the real interesting finding here. So when you look at this picture here, this video, a mouse is being shown a complex scene. So it's not just a simple round spot, or a blue sky. It's a complex picture. And so a lot of nerves have to come together to understand a complex feature, what Bijoyji was saying, the individual elements, the features of a person's face, a person's gait, and so on. But what was interesting is, even though they showed that mouse a complex picture for a certain length of time, very quickly, the mouse stopped paying attention with all of its neurons on the visual cortex to all the elements of that picture. And so what that showed was that there was something happening in the subcortical structures that determined whether or not the visual cortex needed to spend any energy on looking at the scene anymore. Meaning the subcortical areas of the mouse's brain decided, okay, I've seen enough. And what is enough is the question that we are pondering. What is enough for survival? I have looked at this picture and there's nothing here that's a threat to me, and so I don't need to pay attention to it anymore. And it ultimately boils down to, do we see only to see what does it mean for me? And I will touch on this question as we go further. So is that an underlying algorithm? Are we engaging with the world purely to answer the question, what does it mean for me? But what we do have is evidence that there's a power law in effect. And that means that it's hardwired and it's probably present in humans as well, though no one's yet repeated this study in us. And then we have the other finding from the University of Parma in 1992 called mirror neurons, Dr. Rizzolatti and his team. So we have the visual cortex on the very last right side of the brain in this picture, which is the occipital lobe. But when you take a monkey and you put electrodes in the brain of the monkey and you show the monkey a tray with a raisin on it, and either the experimenter picks up that raisin or the monkey picks up that raisin, the areas of the cortex shaded in gray light up. And that was an interesting finding. It's not the visual cortex lighting up, it's all the areas that relate to motor action, planning a motor action that light up. And planning that motor action lights up, regardless of whether it's the mouse moving its arm or the experimenter moving its arm. Hence the term mirror neuron. Dr. Rizzolatti's paper, original 92 paper, didn't actually coin the term mirror neuron. It sort of came out in the literature afterwards, but it's become the standard term for this phenomenon, mirror neurons. And it's been shown in humans, it's been shown in lots of species, so it's evolutionary conserved. That's important. We have to deal with it. We have to incorporate it into our concept of how the brain works. The brain works using mirror neurons. So what is a mirror neuron? A mirror neuron is something that acutely, actively, in real time mirrors what is going on in the outside world, but it's doing that in areas that are engaged in planning for action in response. So that was a very deep finding there. And then we have the extrastriate body area, which is the picture on the left side. So we have areas of the brain that light up, and all of this, in humans is thanks to the development of functional MR imaging. So when you tell somebody, twist my arm, right? This area in pink and blue lights up. So these are not exactly visual areas, but they're related to the visual areas, which are right next to them. They're in between the visual areas and the motor planning areas. So whether you twist someone's arm or you tell someone, twist my arm, I'm going to twist your arm, this area lights up. So as you speak to someone, his or her brain is already mapping that speech into action. The brain is visualizing physically that action occurring in an effort to see how it should respond or what it should do in response. So that was also a very new finding and very interesting finding. It doesn't have a catchy name yet that I could find, like mirror neurons, but it's there, extrastriate body area finding. And then we have objective face trustworthiness. So the reason for the word objective is because if you ask someone, do you trust this person's face? Invariably, they're polite and they say yes, but their brain may disagree. And we have, 30 years of data now on this. What constitutes a trustworthy face? What constitutes an untrustworthy face? And apparently it has something to do with shading around the eyes. And so there's computer programs that you can buy that make fake faces, and you can increase the shading or decrease the shading and make the face more trustworthy or less trustworthy. But the question that was interesting for us for this talk is neural clusters. Where does this processing happen? And it turns out, again, just like blind sight, just like awareness during hemi-neglect, it takes place below the cortical level. So processing of face trustworthiness bypasses the visual cortex, just like a lot of other things. Also very interesting is that untrustworthy faces are better remembered than trustworthy faces. And, Bijoy mentioned the word survival. I think, it's better for your survival if you remember an untrustworthy face. So this seems to support the idea that survival is important. And then the French and the Chinese collaborated on a study to see if there's any impact of different ethnic or cultural facial features on this objective face trustworthiness, and the answer is no. whether it's the French looking at a Chinese face, or the Chinese looking at a French face, or the French looking at a French face, or the Chinese looking at a Chinese face, the mechanism of determining face trustworthiness is the same. So then we have this other thing, we've already moved from thinking that long-term potentiation was the mechanism of both language production and memory formation. We have now a large amount of data on something called non-synaptic communication or gap junction communication, which involves small proteins. These are peptides that cross from one cell to another. So you have these two cells, one on top, one below. And these peptides trigger various receptors like G protein coupled receptors and other receptors to immediately create other proteins or immediately cause a calcium spike and to immediately create changes in the nucleus of the receiving cell. These neuropeptides can also be transferred over long distances through something called exosomes. And that creates another way of non-proximal conduction of information from one area to another, which is slower than synaptic conduction, of course, but it's not anatomical. So there's no clear nerve that you can follow from A to B, which makes it very challenging to discover what the intended recipient is and how it gets there. So there's a lot of people looking at this, but it exists. And if we are going to think about how the brain understands the world, it's something that we need to incorporate. A memory engram, therefore, may be independent of synaptic potentiation. I think the data is really persuasive on this point. So an engram, I think, can no longer be credibly understood as just an increase in dendritic connections in a nerve, or an increased number of neurotransmitters inside vesicles in a synaptic gluton. So they're a form of long-term storage. Long-term storage requires the least energy possible. And so the speed at which we pattern match, which is unmatched in the universe, the human brain is the fastest at pattern matching. So that speed, like I said a year ago in the audio talk, speed is something we have to take into account. We pattern match very fast, very quickly. Therefore, not only have we taken in the information coming in, we have compared it to something that is stored in us, and we are doing that within 20 to 30 milliseconds. That's fast. And so we have to think of what possible mechanisms allow for such a thing. And Bijoyji mentioned proteins, energy, and that pushes us into considerations of energies and bonds and energy states. And that's what these drawings here are. Energy, which makes proteins fold in a certain way to have the lowest energy state, happens within 1 millisecond. And Dr. Pandey very kindly allowed me to show this video here today. This is a video that he made in 2010. I think it's an awesome video. And the whole thing is 1 millisecond, everything that you see. So you have a helix forming on the left side, an alpha helix. And then you have the other, on the left side, by the way, is a C terminal, on the right side is the N terminal. And you have the other areas of that protein trying to find its lowest energy state. The lowest energy state here, of course, is the beta sheet. So the beta sheet is trying to form, but it's stuck for some reason in a non-native trapped state. So it has to figure a way out of that. And so the impetus of having to, of course, end up at the lowest energy state forces further conformational changes. And eventually it arrives at its final state. And you'll see that the C terminal is still sort of loose and not quite where it's supposed to be. It'll get there. And the reason it gets there is energy. So energy drives all of this, and all energy drives all of this within a millisecond. So anytime we think about pattern matching, and we think about engrams, and we think about memory and recall, instant recall, we have to think that the synaptic long-term potentiation route probably is too slow to account for it. And so people have been looking at what possibly could be other mechanisms. So one of those is the Glansman team at UCLA. And so some years ago, they proposed that all these memory engrams are stored in DNA. And I was happy to see this picture because that was what I also eventually thought that's where it must be, because everything is stored in DNA. 80% of our DNA we've been calling junk DNA for decades now. And it's probably not junk, because anything that's evolutionarily conserved is not junk. And so we may be storing all of our memories in DNA. So it's not that hard to visualize. It's like an audio tape, like a magnetic tape. You have a tape head. And so we have tape heads in our cells, and they read these tapes. And so it is possible that long-term potentiation, which is a fact, may play a role in the immediate storage of memories. In 94, I actually met Dr. Bujaki at a science conference in Budapest, and he played an audio cassette. And the audio cassette showed in the form of piano tones, ting, 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 how the hippocampus of a rat was storing the day's events. And he was able to show that we store memory from newest to oldest. We play it back in reverse. And so, the possibility is that we don't store that memory in electrical form in neural connections or dendritic connections, as in pure anatomical storage, but it's actually stored in our DNA in the form of nucleic acids, which is a very exciting concept. But again, it's a concept for now. So we also have evidence that, as I mentioned before, untrustworthy faces are stored better. So there's this algorithm that is in effect in all of us automatically. We cannot stop it. We don't most times even understand that it's there. Like Bijoy just said, most of the time we live in the gross world. We don't pay attention to what our brain is actually doing or telling us. But untrustworthy faces are proven to be stored better than trustworthy faces. So this algorithm, in effect, plays a role in every type of functioning of the brain. So this is where I was talking about. We started with long-term potentiation, which is synaptic gap junctions between two cells. But now we're also looking at field potentials and electrical potentials that are created at the other membrane. That's the membrane between the nucleus and the cytoplasm within the cell. And it turns out that we actually have these nanopores in the nuclear membrane where DNA, double-stranded DNA, is unwound, and sections of it are clipped and sent out into the cytoplasm as either single-stranded DNA or single-stranded RNA. These are what are called non-coding RNAs, meaning they don't code for a protein, but they must be coding for something, because again, this is evolutionary conserved. So they also generate a calcium current at the same time. So not only is this piece of RNA or DNA, this clip coming out into the cytoplasm, it is coming out at the same time as a measurable electrical current. And this, again, puts us in sort of the time scale that we are thinking about, something that happens very, very quickly, something that happens that is related to energy, and the speed at which energy and bonds operate. So this is all very, very new. We can't say anything more at this point, in terms of incorporating it into what we think of a neural cluster, but I wanted to show you this today just to show where things may be proceeding. And then they've also been able to transfer memory by syringe, which is a very interesting finding. So this is a sea slug, the aplasia, it's a worm. And they fear conditioned one aplasia, and then they took the cell proteins from that aplasia, the RNA from that aplasia, and transferred it to another aplasia. And that aplasia, which had never been fear conditioned, behaved exactly like a fear conditioned aplasia. So the information was transferred by RNA, which is another, I don't know, you could call it direct or indirect evidence for memory being stored in DNA or being transferred from the nucleus in terms of pattern matching, but it's suggestive. The other thing that we have to think about is the Greeks said that the object drives the brain. Information about the object drives the brain. The brain is this blank slate. It knows nothing. Everything has to come in from outside. But I think the evidence is undeniable at this point that if there's an active interaction with the incoming information, and there's an active intent. So when the brain is dealing with incoming information, it is dealing with it with an underlying intent. And that intent is, what does this mean for me? So it is not a neutral passive observer. There's an active participant in this interaction, and its interest is to see, what does this mean for me? And so what we see is what our brain lets us see. So in the lower picture is a picture from Dr. Andrew David Hussey, who is at RTI. So on the left side is a picture of a flower as we humans see it. On the right side is the picture of the same flower as bees see it. So what is objective reality? Objective reality is whatever your brain shows you. And you see that also on the right side with these Ishihara plates, where if you have red-green color blindness, your world is kind of different from people without red-green color blindness, but it's your world. This is the only world you know. So what is objective reality? Objective reality probably doesn't exist. So the Greeks are probably not correct on this point. So we have here also from the Rizzorati lab back in the 90s, this very interesting experiment where you have this monkey and they have the electrodes in the monkey. And what they're doing in this experiment here is a gesture. One experimenter is giving a piece of food to another experimenter. Monkey is watching. But what is the brain of the monkey doing? The brain of the monkey is interpreting that gesture again in terms of, what does it mean for me? So the premotor areas light up, they spike up, and those are the areas where the motor engram is stored, meaning the actual mechanics of how to move, right? How, if I want to touch my nose, there's an engram in my brain, in my cerebellum, that tells me how to touch my nose. In fact, 90% of it is in the cortex. The last 10% is in the cerebellum. So if you have people with cerebellar disease, their finger will come up to here, and then when the cerebellum has to kick in, it goes off. So there's these preset engrams in the premotor cortex of brains, all mammalian brains. So what the monkey is doing when it is looking at one experimenter handing a piece of food to another experimenter is deciding what actions it may need to make. And that was a very interesting paper, very interesting finding, and it's been repeated a few times by people who are able to do this. And it's also been now shown much more easily with fMRI. So putting all of this new information together, how do you synthesize a different or a newer concept of neural cluster formation and the actual dynamics of it? The dynamics of it is still something that we have to think through. The thing that we can say safely without speculation is that they are not passive reception clusters, meaning that they are not echoes of something that is sent in by the external object. They are active, participating clusters that aim to give us some usable, actionable information. And so this need for preparing for an action underlies every interaction with the outside world. Neural clusters are both geographic and temporal, meaning the Hebbian concept that neurons that hang together, fire together, they may not hang together, but they can still fire together. And they may fire together in different areas. So for example, when the monkey is seeing something, its visual cortex is not lighting up. The motor cortex is lighting up because it's planning, what should I do? Then they're widely dispersed in brain areas. What does that gesture mean? So we are constantly, actively, in real time interpreting everything that we are seeing. And by we, I mean our brains, whether we are aware of it or not. And that was one of the things that Bruno and the others explained, that the top cortex, which is what we are mostly aware of, may not necessarily be aware of what's happening in the subcortical areas. There's a lot of automatic processing occurring in the subcortical areas that plays a direct role in everything that we do 24/7, and seems mostly aimed at self-preservation, survival. So that's about all we can say from the current state of the data, because everything else would be sort of hand-waving. But we do have some idea now of how to think about this thing that we call a neural syntax. What's a neural syntax? The syntax is what the machine uses as its language, the brain language, the machine language of the brain. And we have some idea, some evidence that the algorithm employed is automatic, applies to everything, and most likely tries to answer the question, what does it mean for me? But we still have no idea about the mechanics of how that algorithm operates. We do know that it operates automatically and quickly. So it is something that requires a lot more thought, a lot more experiments, a lot more modeling. So one of the things that we have to incorporate into this modeling is something that explains the short time scales. And so we have to think of various ways other than long-term potentiation that can explain how these things happen so quickly. So in summary, the anatomical neural cluster has been, I think, safe to say, superseded by the concept of dynamic clusters. So dynamic, not only in terms of within the column, but also across the brain. So a dynamic cluster is an energy cluster. You can measure it with electrical tools. You can see it on fMRI. And it's a form of energy that interacts with your brain in order to reach a decision, an action decision. It happens very quickly, even before your surface consciousness is aware of it. And I think it has to do with answering the question, what does this mean for me? Because that's a basic survival question. And so that's presently where we are. There's more unknowns than knowns. Thank you.
People can unmute yourself, and can you have a round of applause? Unmute.
Thank you.
Unmute. Okay.
Excellent.
Go for it.
Very good.
Most
Go,
kind. Thank you, Ji.
go for
Very
it.
good presentation.
Go for it. Boy, all right. So, okay. So, did all people applaud? I think again, I must say that this is a large survey of difficult information orienting towards an hypothesis to determine how objective the brain is. Is brain a tool or brain the truth? So we are of the opinion that the universe rests in our brain. It creates the universe for us. And it is intelligible to us, except we don't want to understand it. It is not understandable directly by analytic means, but it can be understood through meditative means. See, that's what India discovered thousands of years ago. We do not know how they discovered, but somehow we're trying to figure out if we can create some science out of it. The subtle information called buddhi does exist, probably exists equally in all of us. It is a protection for the individual, for your life force, to protect you, not against, because of the universe. What does the universe do? It's not trying to attack you. It's trying to load you with stimuli, information, such that your brain can be saturated, such that your thermodynamics of the brain can destabilize, such that you can have a different temperature. And that happens to us. We jam up, we get confused. So that's the empirical observation. So the question is, so then the brain has the automatic mechanism by which to release that information out. So those are the motor functions I was talking about before. That's the hypothesis. That is a built-in motor in the brain, which dispenses the stimuli energy that comes to the brain without being asked for. The sun shines, the brain has to accept it. So it could be there for your biological reason. But if the sun shines on the brain too much, brain gets hot. So the brain has to dispose. I kind of wipe my head and so on, get cool, things like that. So let us open for questions. People feel free if you want to research on the topic or around the topic, about the topic, you can say a few words about it. And then you ask a question. We'll try to answer as much as we can. As we are reported to you that we have been trying to find funding agencies who might support us. People are not very friendly. Not friendly means people don't understand. So we're gradually creating our own language so that we can communicate. We have not even written a whole manual about it. We will do. But let us see. Let us have a good half an hour Q&A. So feel free to any question that you may have. Raise your hand or just introduce yourself and ask the question. Anybody has a question? Yes,
I have
Jaydev.
a question.
Yeah, introduce yourself and ask.
I am Jaydev Dasgupta.
and I live in Southborough here. I'm very familiar with the IDC program, and I've heard a few of these seminars in this series. All of them are very, very interesting and informative. The question that I have is that Dr. Padmanabhan made a statement that objective reality probably does not exist. Okay. So what does that mean to a brain scientist when he looks at the brain? Is he studying the brain, or is he just his brain is telling is something is an artifact of the brain? So when we look at it that way, I would like to hear your thoughts on that.
Well, let me just interject a little bit. I think what I mean, he will say, but what basically what we are phrasing it, that's not knowable by us. Objective reality doesn't exist. It could be existing, but it is not known to you. You have no access to it because of your grossness of your operation. And if you do indeed go to understand the object reality, then you suspend yourself totally from your cognitive functions. Hence, you can't bring it back.
So
So, I
that's good.
understand it. So, this is kind of a uh, science of it we're trying to understand. But Bharani can say.
Yeah, please.
Right. So when you are a scientist and you're looking at a histology section under the microscope, you may think that you are looking at objective reality. After all, you sliced this with a microtome and it's fixed in wax and it's right under your eyes and you're looking at it and it looks crystal clear and it's crisp and you can see this and you can see that. But what you are seeing is what your brain is showing you, right?
Obviously.
Yes. So, if you think you're looking at a red flower, you think it's a red flower. If you think it's a gray flower, it's a gray flower. So people who are colorblind, until they realize and they learn that they're colorblind, do they realize that they are not looking at objective reality? No. So what is objective reality? Objective reality is different for different people. It depends on what the brain is showing you. And the point here is you have no way of knowing what that objective reality is because you have no access to it other than what your brain is showing you. And the brain is showing us a lot of stuff that we basically ignore. For example, face trustworthiness information, so on and so forth. Most people, they just ignore it. So there's stuff that the brain is showing us, and there's stuff that the brain is not showing us. And so long as we recognize that this fact exists, it allows us to appreciate that what we are seeing is what is being shown. There is no denying that you are looking at a nucleus or the endoplasmic reticulum or, cell membrane or some such thing, but there may be different beings that see those differently. So it's not as if the same yellow flower appears as a yellow flower to everything that's alive. So the definition of objective reality is what we are wondering about. So what our reality is our reality. And by us, I mean all humans. All human brains see things the same way. They say a dog sees the world in black and white and gray. I don't know because I'm not a dog. The objective reality for all dogs is different from the objective reality for all humans.
Yeah, I think Sibaram is here. Sibaram just sent a chat message, and which is very important. Let me share that. What he did just a thesis. Can we all congratulate Dr. Sibaram? He has been working on Vedas and so on, and he just submitted thesis. So let us just applaud his efforts. So what he said is that the amount of time that we give to understand is not enough. So not that you may poke through it longer and longer and longer and then analyze this deeper and deeper and go to those higher scales in the brain and to see, do I understand it, how they connect and this thing. But we don't. you get some superficial information and you get going. That's his point. So I also appreciate that he said so. Um, okay, other questions?
There is a question by BVK Shastri in the chat
Yeah.
that says, please explain the term brain language and machine language model, which I think, Bernie, you talked in the in your discussion.
Right. So the neural syntax, I thought, represented a language that the brain is employing in order to answer the question, what does it mean for me? In order to understand all the particles that are coming in order to first create all the particles and then put them together in a way that will lead to an action decision and to do it very quickly. So there is an underlying algorithm. I think it's pretty clear now that there's an underlying algorithm in effect that operates in all spheres, visual, sensory, and so on. And it operates using power laws. At least we have some evidence for that. And so it's a physical manipulation of information within the brain. And anytime you find manipulation, it follows a plan. And I think the plan is, what does this mean for me? But how that is implemented, we have no idea. But we know that there must be some plan, which we are now sort of thinking of as a neural syntax, the syntax of the brain. We don't have the Rosetta Stone, for the brain. So before the discovery of the Rosetta Stone, nobody knew what all the Egyptian hieroglyphics meant. You needed a translation device, and we don't currently possess a translation device for how the brain creates those particles, how the brain selects between those particles to choose a decision, how it stores those particles. Bijoyji mentioned at the beginning, how do I know this is my father? Is it the shape of his jaw? Is it the specific features of his face? Is it the way he walks? And of course, you've known your father all your life, so he's not the same guy now that he looked 20 years ago, 40 years ago, but you still know him as your father. So how does the brain break down these elements? I haven't shown a slide for that here, but there's been some work on coupling, the wave coupling, brainwave coupling, the phase of the coupling. And there's been some work done to show, for example, you show a picture of a baboon. How do you know it's a baboon? So it turns out that, if you actually carefully measure the phase of the different brain waves that are generated when you show somebody a picture of a baboon, depending on the phase of the coupling, it may or may not, or I may relate to a particular feature of the baboon. For example, the shape of the face or the shininess of the silvery fur and so on. And so it's at this point, we don't have enough to say one way or the other. All we can say is it most likely exists.
So first let us acknowledge the presence of Dr. BVK. He connects from India. He's one of the very first few scientists, scholars who thought about this language problem, language in the brain problem. He comes from a Sanskrit background, so he tried to think that whether what we say, what we call the sounds, do they mean anything? And he has been thinking about it. And maybe he will explain a little more about his research. But regarding his question and regarding our current discussion, like machine language versus brain language, what we learn today and what we think we must understand very well, the brain doesn't have a language. that we think as language. So the brain has this organization. Brain has a massive network of neural organization. Most of them are transient. Most of them are plastic. Most of them are recombining. So the question is, what are the units of them? Whether I see a forehead is a unit, or this chunk of my face is a unit. How does the individual information content, I call particle in the beginning, so that unit and hydrogen atom of the brain has to be somehow understood, that this is the information quantum. Now that quantum is not discrete. That quantum, also like any nuclear structure, is analog. It smears. So it is not that it is a structure, except when a signal comes, it manifests depending upon where it is from the phase of the brain, state of the brain, and the protein that it contains. So this analog brain structure eventually somehow translates into our speech, which we may call language, which appears discrete. But that discretion is not due to the brain, but because the muscular structure of our organs, of our vibratory systems. So the machine language as discrete different words or structures or something, which we say this separates that and this thing, as the concept of language happens in our oral apparatus by virtue of vibrations, not at the, we do use a term lately we call proto-language, but I don't think that language, this language. So when we say brain language, we have to fully understand that it is a connected, smeared, massive organization which is ever changing, which is floating, which is recombining and creating new concepts all the time. But BBK, you can tell about your own research a bit if you like at the present time.
Okay.
Are you there?
Okay. Thank you, Mr. G, for all the good words. I have been just a baby. Yes, I'm here. Can you hear me?
Yeah.
Can you hear me?
Yes, of course. Yeah.
Okay. So thank you for all the good words. I'm just a baby in this field. The problem is, right now I'm in Thailand conducting a workshop. The workshop is just like this. Say a sound. It could be om, it could be tum, it could be gum, it could be any particular sound. No meaning associated with that. Repeat that sound, observing the sound beginning to end and the gap that follows as a silence, and repeat that sound 3 or 4 times. That is the experiment. And the subjective experience obtained by a batch of 10 persons, okay, is entirely subjective and different. One feels relaxation, one feels heat, one feels the energy going up. I mean, their expression of the language is so diverse. That is not the point. The point I'm trying to highlight is a conscious intervention of a sound triggers brain-body reactions. And that varies from person to person in the context. So do I understand that language as we understand in the traditional jargon as bhasha, chandas and all that is something? Do I understand the language as a brain language, which is a biota, biomatter, kind of a structure, or do I understand language of the machine as a structured programming with alphanumerics? This is what I'm trying to get a clarification.
Okay, no, I think very good point. Very good point. Not that we can answer this, but you did hit the point very clearly. That is what happens when we are hearing, what you are hearing me. So the amount of
when you say the sound
intake that comes to 10 of us from the signal, is it same for all? Is different. So the question, and you say it is different, and that's exactly a fact. But then question is, so what Bharani presented today, that by virtue of our habitat, the way we operate, way we see our local universe, local world, family, trees, system, so we develop these engrams in us, which are patterns that we recognize quickly. So which means that suppose the person, you speak to him in his language, her language that he is familiar with, he probably will catch faster than something else. Now the question then you said that in that process, how does the brain operate? He understood it. But then question is he happy about it? Whether the brain, we talked about truth a little a while ago. So the brain, does he say, did it understand that now it is my mother's name? I love it. I have a lot of respect for my mother's name. But does that give him total joy, or it's another piece of knowledge? So what we are saying is, or what we discussed before, that this ultimate brain's function is a total connection to the universe as joy, where there is a resonance, that I feel one with it. That object is me. I lean myself on that. Or I understand that as you, as another object. So that thing we have not fully reconciled yet. I'm of the opinion, or I feel from my own experiments, that there's a merger. And that merger can happen. We do not know who causes that merger, whether we cause it or it is a signs of it. That is, the signal itself becomes so unified that brain and signal become together. That a BVK becomes me. It's kind of imprinted in me. So you are not another object anymore. So that Bharani told about this Greek object theory and understanding us as subjects, and you are not an object anymore, but we are connected. So that we shall see. I think I'm trying to, the next part is my talk, which I'll do. I don't know when I'll do, but hopefully I'll do, try to expand on this and see the science of grammar and science of expression and science of understanding through the neural theory.
Okay.
And
Just
so,
one more question, one simple clarification required.
Yeah.
When we talk of this whole neural process, my understanding is it works at the speed of whatever the electricity or the biosignals flow in the body, which is proximate to the speed of light. Do I understand that correct, or is there a difference?
Well,
Bharani.
I said that a year ago, but it's not entirely a universal consensus that our nervous system operates almost at the speed of light. But over this past year, I have found nothing to dissuade me from that view. Because we have to have some mechanism that accounts for 20 to 30 millisecond response time. And since the weight of data accumulating seems to show that we need to move away from this exclusive synaptic communication, long-term potentiation sort of model, which Glanzman and his team basically had just flat out called a hypothesis, right? After 100 years, it's still a hypothesis, the synaptic hypothesis model. So the answer is we don't know. You know, for years people thought they knew, but it's now pretty clear that it probably wasn't correct. So we don't know the actual speed at which the human nervous system operates, or the mammalian nervous system, because they seem to be operating fairly similarly. But the evidence seems to be showing that it's faster and faster than we thought it was.
But I think you should, I think you probably know that BVK, that is something has come up in the literature very strongly, that depending upon what you are, you know, you have heard this word called pratibha in the Bhartrihari literature, and we call buddhi in the Sankhya literature. So depending upon your application of it, depends on how much of the signal you need to frame the image. To make you as a person, do I need 2 pixels, 1 pixel, or 3 pixels, or to see you all through? So that Bharani came through, trustfulness, that kind of concept, that how much I must see before I recognize. Or I do see a stress of it, I recognize. I see the Lord's face from his feet. You know, I mean, depends upon how much affection you have to the object. So that kind of a concept, in the sense, what it takes you, the universe may feed you the whole lot of bandwidth of signal, but you may need only a small 100% of it to really phrase it. So that the brain does, because it has the prefabricated networks, templates, engrams, and then the question is how quickly we access it. So that's the pratibha part of it. That is whether how much of clean
Do
we
I have
are.
time for one more question?
Yeah.
Do I have time for one more question?
Just hold on. If anybody
Okay.
else has, then you come back.
The case study is
Yeah, let us see
Okay.
what anybody else has a question. Then we. This is very interesting. So you are one of our brothers in this tradition, because we are not. See, we're neuroscientists, but at the same time, we have been trying to understand the so-called. You know, I tell you where the this this thing about the differentiation of the brain, of the gross and subtle, not academically, but physically, came in. I was reading Ramayana and trying to interpret that for children and trying to make some translations. And then this guy called Sugriva, so he tells Rama that Shukma Buddhi Bicharaya. So the question is, so Rama is thinking about my Sita, my Sita, my Sita. So that doesn't do it. Do it like how best I can reach there. Sukma, step by step, strongly. So those kind of things brain has, we don't accept them. The success of an event is step by step. So those steps are known to the brain. So anyway,
Bijayji, Sivram
go.
has a question.
Yes, Sivram, please do.
Yes, thank you.
Congratulations for your successful thesis.
Thank you. Yeah. So my question is around when we talk about neural cluster formation and how they interact with each of them. Is this more dominant with respect to the cortex, that's visual signal processing? We spoke about subcortical structures as well. Or is it also same with any other reception of sensory signals? For example, auditory signal processing also has a dominant clustering, or is it specific to cortical clustering?
So neural clusters are, as we define them now, neural clusters are independent of the modality of a signal. They are, as we see them, not even connected to incoming signals. Neural clusters also form in response to memories.
Okay.
And so a neural cluster is a piece of information that is presented to the brain in order to arrive at a decision. And a neural cluster, that information, however many neurons are involved, that's how many neurons are involved. It's a dynamic process. It's an energetic process with a goal in mind. It is not just simple entertainment. So the cluster that is formed is an idea and a piece of information. So whatever many neurons and wherever areas of the brain come together to form this particular piece of data, we are calling that a cluster. And there may be small clusters and big clusters. Some piece of information may require a few number of cells. Some piece of information may require hundreds and thousands. And different areas of the brain come together to form a cluster that provides some actionable information. So it's diffuse and completely independent of any sensory modality. So when you, for example, if you look at that twist my arm data, it doesn't matter if you're seeing somebody twisting an arm. It doesn't matter if you see somebody twisting your arm, or if you hear somebody say, I'm going to twist your arm, the same clusters form. And that was a very interesting finding. So basically a cluster is anything that a brain needs to decide on an action.
Okay. Thank you.
Sadri.
Unmute, Ram, unmute yourself.
Yeah, really nice talk. Thank you very much, Dr. Bharani. Going back to the objective reality, I just want to make one statement. It really depends on where you place the observer. If I think of myself as body and mind, if body is the observer, then there is plenty of objective reality around us. If I go touch a stove, I'm going to get burnt. There is objective reality, right? So the way I see it, the brain evolved to overcome the uncertainty, position and data. And the position part is observation, which comes from the body sensors and the brain portion that is connected to that. Whereas to get the continuity and to see the velocity of change, the mind portion of the brain evolved, and that is the deductive part of the brain. So everything you deduce with your mind, yes, it is real only to your mind. Whereas if you drop something, it's going to fall. That is objective reality. And touch something hard, you're going to get burnt, and all those things. The body portion of the input is real.
It's real for you, but that's the thing.
No, but the brain is as an interface between body and mind has two portions. When you say subcortical, we are really referring to the portion of the brain that is talking to the body organs, the sensory motor organs. Whereas the mind is your database that took all your past experiences and converted into your memory database and allowing it to both to learn and to recall. So that exists in a sphere by itself.
Well, the trustworthiness, untrustworthiness data sort of goes against that concept. So and that's why they very carefully use the word objective. So trustworthiness, untrustworthiness computation takes place subcortically. It doesn't really involve your mind. It definitely doesn't involve your awareness. And so we have pretty robust data now for about 20 years from various groups everywhere, that if there's something that happens before 350 to 500 milliseconds of you seeing a face, and it is not conscious, that one is very sure. There's numerous game theory plays that they have done to show that the people are not conscious that their brain has done this computation. You ask them why they picked this why you picked X over Y, and they can't tell you. And so, yeah, I don't think it is entirely mind in that sense, or at least you have to probably expand the definition of mind to include these automatic processes. In terms of
What I'm talking about.
objective, the 25 years ago, I got clobbered by a bunch of scientists when I used the term intrinsic worth. I said, what is its intrinsic worth? And they all fell on me saying, what on earth is intrinsic worth? There is no such thing. Something is worth only what others think it is worth. And so what is objective reality? I think it is objective to us. The point that you make that if anybody touches a hot stove, he or she is going to get burnt is true for humans, but may not be true for certain other species. Again, it's what we experience.
So isn't it that all the science and everything that we are doing is all anthropomorphic? So shouldn't we just talk about the world with respect to us and not just talk about how bees sees and how dogs see it?
So that's where the brain comes in. The whole neuroscience says
So
you're
that's what I'm
out of
saying. So
your
it has to be
body.
anthropomorphic,
Yes,
the whole construct of science.
yes. The
And we are
brain is
looking
so the question is, you have a brain science, not the physical science. So physical science is what the brain has given you, and that you operate with. But now you're going inside to the brain, that what the brain is seeing. So that is not what the behavioral science says, that that's the tree and that's Jaidev and this is this, but that's the brain is seeing. So it's not seeing Jaidev as an object. So brain seeing neurons, Jaidev's particles, and then see that whether Jaidev is trustable, not trustable, do I keep away from it? Do I love him? So all that happening inside of the brain. It is not how useful you are, how scholarly you are, or which is growth. So this growth versus that we are trying to understand.
So
the subcortical brain is part of the brain. I'm sorry, subcortical brain is part of the brain. You cannot think
right.
that
but when you said that it's different from the mind, I think it's you can't really say. So if you look at trustworthiness, is that mind or brain?
If you don't, mind is a debatable term. Let me call it the cortex broken into a mental cortex and a physical cortex. Physical cortex is connected to the body. So that is the peripheral nervous system going through your spinal cord and the brainstem and the midbrain. It is part of the brain. You cannot ignore it, which is, when you say subcortical, you're making it look like it is not part of the brain. The whole thing is, if you want to call it a nervous system, it's a nervous system that helps you get rid of your whole uncertainty, help you understand the whole thing. So there is objective reality on the outside.
So another point that Bharani I have is that if the subcortical part, let's say the subcortical brain differentiates between trustworthiness and untrustworthiness, right? That's what you said, without having the conscious mind or the top layer coming into picture. If the subcortical, is it possible that the subcortical itself has access to the memory domains where the person was found to have or determined to be trustworthy and non-trustworthy? And the top layer does not know that, but the subcortical brain has that access.
There he brings the RNAs. We have not studied that yet.
No. It doesn't involve memory. So even so, this algorithm operates even when you have never seen that person before, and that algorithm operates in order to protect you from strangers. So the algorithm operates to help you decide whether or not I trust this person. So it has nothing to do with memory. It has nothing to do with stored things. But if you come across someone who proves to be untrustworthy, there's a very strong memory engram made, so that the next time you come across that person, your brain is hammering you saying untrustworthy, untrustworthy, untrustworthy. And that's been shown.
Okay, all right. 11 o'clock, so we close now. So anybody, I think you are all Does the man have anything to say?
No.
Okay, all right. So anyway, so thank you all. This was our
Thank you.
fourth. So the next one we'll try to see to logically connect all this to the motor functions and the language. I'm working on it, and if it works out, I might do it in December, or it has to wait. It's a function of classification of the motor functions. The logic, the model that we are trying to build is that the stimulus as it comes, Professor Mohanty is not here, I think I mentioned that in the beginning, that the stimulus, when it gets disintegrated into these neural tokens, and then you have to, which organism or how best they get organized, reconstituted. So that is Bharani's point that it's a cell protection. So what is cell protection? Cell protection of the individual motor functions. How best I kind of come out of this, whatever stimulus I'm invaded by. So question is there a hierarchy of the motor functions, their energy levels, the way they dispense them, way they combine themselves together with, and how the disposal of energy will take place, the passageway of, so which means as my hand is operating like this, it dispensing energy that is inside of me. So why hand, why not leg? Why not? I understand the eyes happen and so on, but anyway, all those from the language perspective. Why language? Because we think language detected the motor functions before. And hence we can take some clue from there that this is how the motor is functioning. It is hypothesis. It's kind of very nebulous. But I'll try to see if I can make a coherent story about it. And then I'll talk to you, hopefully in December or early spring. So we shall see. I thank you again for helping us navigate this channel. And as Ram rightly pointed out, so this is kind of a controversial, but we believe that controversy is because the world is created that way. The world is created that objects exist and we see an object. So when we turn that back, that I make the object could be there, but the object in me is what I created. So that's the difference. And we do not know why the world did not start that way. And we think that's because the brain as an active participant in the object processing was not known. Only eyes were known, eyes were seeing. But eye gets the data, the brain processes it. So that whole processing, it could be discarded because too complex, or it's not necessary for our normal operation. Just like Ram said, the fire is hot, keep away from it. But that's not a science there. That doesn't mean that you understood it. You have to burn your hand in the fire to understand the strength of the fire. That's science. So that is, then you can say, okay, this fire will burn my finger up to half. Then you quantify it. It doesn't burn this. It can burn that. It can melt this. We see that, myself and a friend of mine, he's not here, we are studying these Vedas these days for the last 5-7 years to understand how people are making empirical science by doing forest fires, by making understanding it. How does the wind blow? How many leaves it blows? Is there a hierarchy of wind power? So how do you know that? You don't say the wind comes, I go away. You study it, understand it. You have to stand there. Does it blow me away? Okay, now it displaces. I'm out. Okay, how much water rises? So things of that nature. We shall see. We shall see. Thank you again. Thank you again. It's a great day. Have a good walk and so on. Winter will come.
Thanks, Bharani.
And we shall see. Thank you very much.
you.
So can we have an applause for Bharani at the end?
Bharani,
Aw.
excellent.
Thank you, Ji. Thank you, thank you.
Great.
Thank
All
you.
right, very good.
Namaskaram.
And thank also Prem. He helped us with the processing everything today.
Namaskar, Prem ji. Thank you.
Namaskar.
Namaskar.